Director, Procter Gamble, P&G International Operation in Singapore. Yeah. Okay, so yeah, you may wonder what this topic is all about. Yeah, as you know, yeah, everyone is having a consumer products which range from cosmetics, household products, etc. Of course, you would like to ensure the safety of the product and how it will impact for your everyday life, of course, right? So, yeah, before it launches to all the consumers, they require what we call a risk assessment, right? So the risk assessment, what is it inside? Yeah, hazard identification, dose dependent assessment, exposure assessment, and risk characterization. So this require an approach, of course, right? So, right, as you may all know, right, all of these approach consider the understanding about the toxicity of the chemicals, what is the safety concerns, yeah, of the said chemicals, yeah, and how to assess, yeah, in terms of risk to the health effects, yeah, under exposure conditions, right, for the usage of the product, right? After you get all of this risk assessment, right, then we will do the risk management, right? So this management, of course, will impact on the decision-making process and to determine how to best address all of these issues, yeah, socially, culturally, legally, politically, or economically, right? So we need to communicate. In order for us to do this effectively, we need to communicate different information to the interested parties, right? To the government, right? To the consumers, right? Or to the communities, right? So these potential risks and risk reductions, right? So how are we going to manage it, right? In the corporate level, right? And how are we going to mitigate, yeah, if any of such condition occurs, right? So here, our current talk will present about the safety assurance of the consumer product by applying a risk assessment-based approach, right? And employing risk management measures and communication among the stakeholders, right? So today, yeah, we will have our respective guests, right, which is a principal scientist, and you also produce publications on the regulatory about the safety assessment, yeah, and concern and toxicology. So it will be a very valuable lessons, very valuable experience that he can share towards all of the audience here. Okay, so all of the question and answers, yeah, can be asked, right, either by live, right? So we will pass you the mic and you can speak. Yeah, do not use the one on the table because it will not connect, yeah, to the Zoom, because we are doing it in hybrid mode, right? So I will pass you the mic and you start talking, right? And for those who are watching from Zoom, right, you can type in the question and answer to the chat box provided. Okay, without further ado, yeah, I would like to invite Dr. Seok Soga Kwon, yeah, to initiate the talk. Yeah, please.
Thank you. So you have several slides. We can skip this one.
Okay.
Next up.
Yes. Yes.
Okay, thank you for nice introduction. Today's talk, this is my outline of my talk. The first, I'm going to introduce you about the risk assessment principle, and then I'm going to move on to what's the safe range. And then I'm going to give you a real example how we conduct safety assessment for consumer products. And then I'm going to move on to risk management and the risk communication, especially there are safety concern about the trace chemicals found in consumer product at very low levels. And then how we ensure safety of those trace chemicals in consumer products. And then I'm gonna finish my talk with a very short summary. Before I start my talking, I'm gonna introduce our R&D organization. Just watch this video. Yeah, the sound quality. Okay, the sound quality is not very good, but I just want to introduce what we are doing in our R&D organization. And then my job at P&G is ensure the safety of our product. That's what I'm going to talk about today. This is the terminology, which is very important. What does it mean, toxicology? Toxicology, as you know, we are studying the adverse effects of chemical that cause on the living organism or environment. And then in order to study the toxicology, there are two important aspects. The number one is toxicokinetics, and the other one is toxicodynamics. It's very simple concept that what toxicokinetic means is what does body do to a chemical when we expose the chemical. On the other hand, toxicodynamics means what the chemical does to our body. So that is the very important aspect in toxicology. Let me introduce very basic concept about risk assessment. The first concept is hazard, and second is exposure. Let me use in analogy. Let's consider poisonous snake as a hazard. And then there are two ladies. The first one, this lady is close to the poisonous snake. However, the poisonous snake is in the cage. So that means there's no exposure. However, the second lady, she's very close to the poisonous snake, so exposure is very high. So this is very important concept when you understand the risk. So risk is the combination of hazard and exposure. So this is a very important concept. Please keep in mind. So let's go to the very detailed explanation about each concept. The first one is a hazard. So what does it mean by hazard? This one, you can take a look. This is an intrinsic toxicity of a chemical with potential to cause adverse effect or health effect. And then, what does it mean by exposure? Exposure is quantitative measure of magnitude, how much, and duration, how long, and frequency of exposure, how often you are exposed to identified hazard. So that is exposure. And then the risk is quantitative estimate of probability of certain adverse event that may occur in certain population as a result of exposure to identified hazard. So keep in mind that risk is probabilistic estimate, but that is the risk. And then the risk assessment principle means it combines all three basic concepts. The first, hazard. When you work on new technology, and then you need to understand whether this new technology or any kind of new chemical is hazard or dangerous. That is the first step. And second step is we call dose response assessment. That means once you identify the hazard, what's the potency of hazard? How dangerous of identified hazard? That is dose response. And then what is the exposure assessment means you remember that the one lady, she's very close to a poisonous snake. That means exposure is very high. So you need to understand that when consumer use our product using certain technology or new chemical, whether it's dangerous or not, that is very important. But it depending on exposure. So we need to understand that how much and how long and how often consumer will be exposed to new chemical when they use consumer product. That is, we are doing exposure assessment. And then the final step is risk characterization. This means you combine all information and then you need to decide whether there's a risk or not. You remember the poisonous snake? The first lady, even though she's close to a poisonous snake, but snake was in the cage. So that means there's no exposure. That means no risk. Why? Risk equals hazard multiplied by exposure. So that is a very basic concept for risk assessment. If I summarize those risk assessment principle step by step, we call it a four-step process. The first step is hazard identification. If I articulate or further elaborate what does it mean by that is first, you need to understand whether this new chemical is dangerous or not. That is the first step. And then the second step is what's the route of exposure? For example, the same chemical may cause adverse effect depending on route of exposure. Let's take a very simple example, water, drinking water. When you drink water, it's safe. What if you inject the water into your blood vessel? It can be very dangerous. Therefore, the hazard is very depending on route of exposure. This is a very critical concept that you need to keep in mind. And then at the end, relevance to human is very important. For example, when you observe any kind of adverse event in animal or rat, mouse, what if mechanism of toxicity is different from human? So therefore, when you make a final decision on hazard, you need to really understand that whether that hazard information is relevant to human. And then dose response assessment. This is you need to determine the potency of hazard. In other words, you found a new chemical is dangerous. How dangerous it is. That is dose response assessment. And then second aspect is there's always uncertainty. For example, you have a new chemical, new whitening active ingredient. It's new to the world. You're very excited. However, unfortunately, you don't know anything about toxicity of this new active. So you need to test whether it's dangerous or not. How? You cannot test on human. You may need to test on animal or certain experimental condition. Once you generate experimental data, then you need to extrapolate to human. So there's always uncertainty when you extrapolate any kind of safety data from experimental condition to human product use scenario. Do you follow me? Makes sense. And then exposure assessment is very critical, like a poisonous snake. How much and how long and how often consumer will be exposed to hazard. For example, cosmetic products, skincare product, there's a lot of paper published about data we called consumer habit and practice. We know, for example, twice a day, consumer use skincare product, and then approximately 1.2 gram per one application they use. And then how long they use, depending on the person, but it can start from teenager to until they die. It's a lifespan. It's a chronic exposure. So you need to understand those aspects. That is the exposure assessment. And then the final step is risk characterization. We combine, integrate all information to make a final decision. So going back, you have a very exciting new technology. It's a new to the world whitening active chemical. And then you understand it's dangerous or not. And then you understand the potency of toxicity. And then you understand how consumer will be exposed to that new to the world new whitening active chemical when they use consumer product. And then final step, risk characterization. You understand, you make a final decision. Yes, there's a risk, or yes, there's no risk. So this is my daily job at P&G as a safety expert. Any question? Very easy, very straightforward. So this is more detailed information for each step. The first, hazard identification, how we do that, and I can go in more detail later, but for example, what kind of toxicity this new chemical may cause, systemic toxicity or local toxicity, we review all those information. And then many times we also even generate the safety data because there are no existing safety data. And then those response, this is very important because we need to understand the potency of identified hazard, how dangerous each new chemical is. This means we need to understand what's the relationship between those, in this case, exposure and adverse effect. This is those response. And then we understand how consumer use product, and then we can quantitate consumer exposure level to your new whitening active chemical. And then the final step is we can make a decision whether there's a risk or not. So let's move on to real world. Every day, we are using very diverse consumer products. Yeah, these are typical of P&G brand. So what does it mean? In our daily life, we are using many different types of consumer products. What does it mean from risk assessment point of view? This one. Depending on the product type, you notice that the route of exposure is different. Skincare product, major route of exposure is skin because you apply on your face. Pharmaceutical drug, you can orally intake. That means oral injection is a major route of exposure. How about the air freshener? You spray into the air. Inhalation, respiratory system is a major route of exposure. So therefore, in our daily life, we are using many different types of consumer product, which can be translated to, yes, when we do safety assessment, we need to consider what are relevant exposure route for each product. This is very important. And then the ultimate final goal is what's the safe range? When you develop new chemical, new whitening active chemical, this is our ultimate goal. What's the safe range? How much we can use safely in our product. Let me show you video to explain about what safe range is.
Safety guide. Same scientific processes as regulatory agencies around the world. At P&G, your safety guides the ingredient choices we make, so that our products will be safe to use every day, in your home and ours. When we explore At P&G, your safety guides the ingredient choices we make, so that our products will be safe to use every day, in your home and ours. When we explore ingredient options, we ask questions, just like you, and avoid ingredients that can't meet our safety standards at first glance. We simply won't go there. If an ingredient option passes this first test, we define the safe range. Almost everything in the world has a safe range and an unsafe range, including life-sustaining basics, like sunlight, oxygen, and water. Using the same scientific processes as regulatory agencies around the world, we define the safe range for every ingredient we use. So how does defining the safe range work? Scientists test ingredients from very small to very large amounts. The largest amount tested that does not cause any harm is the beginning for determining its safe limit in products. Of course, that amount goes well beyond what we would actually use in a product. So our scientists add a standard, extra-wide margin of safety for even more protection to establish an ingredient's safe range. Next, we determine that an ingredient's use amount in an actual product is within the safe range. If it is, the ingredient can be used in our products. But that's not the end of our diligence about safety. We listen and use a range of experts to assist you with questions and ensure the product is performing as expected. We also continue our diligence by investigating new information about our ingredients and working with scientists outside of P&G. Ensuring your safety in everyday moments of life has been at the heart of who we are for more than 175 years and will be for generations to come. It's our commitment to you and your family, and to our family as well. At P&G,
And what does it mean by safe range? And this is a summary of the video that you just watched. So previously I talked about the risk assessment principle. And then through the risk assessment principle, we identified the hazard. And then during the dose response assessment, we identified uncertainty, because when you deal with experimental data to extrapolate to a human product use scenario, you need to consider uncertainty. And then by applying the uncertainty to identify the hazard, you can establish safe limit. And then the safe range means this one. So in other words, as long as the consumers are exposed to identify the hazard, any kind of dangerous chemical below this level, they can safely use any kind of product containing any kind of new technology. This is the definition of safe range. This one, I just want to show you the overall big picture. So what? So how does safety assessment fit into our daily product development cycle? So this is the typical product development cycle. In the beginning, our R&D people spend a lot of effort to screen out new technology. For example, new whitening active ingredient, and you need to test which candy. It can be synthetic chemical or natural, you name it. You screen out a whole bunch of candidates. During this step, my job is I do risk assessment because I need to screen out any potentially dangerous chemical in the beginning so that we can maximize our resources. And then once the product development moves along, and then of course, we generate further data to further consolidate our safety of new technology. And then if everything goes well, and then we can successfully launch our product in market. And then we do not stop here. We also have a post-market surveillance system to monitor what kind of adverse event happen in the market. And then as needed, we can go back to our original safety assessment and then try to manage any potential risk. This is the whole product development cycle. I can explain more detail later about post-market surveillance. So from now on, I'm gonna go into real daily work, how we assess consumer product. Number one is our safety assessment is based on each ingredient used in product. So as you know, the product, consumer product, cosmetics, or pharmaceutical drug, everything consists of several ingredients. So we need to assess individual ingredient. And then in case of consumer products, we go one step further. We need to understand what's the safety profile of product formulation, which is a mixture of several ingredients. This is what we do. So this is a very simple diagram showing typical consumer product. Here's the formulation. Let's say there are 4 ingredients in this product formulation. And each ingredient, which is raw material, consists of several components. It can be active, or it can be solvent, or preservative, or impurities. Our job is we need to assess all components to ensure that this product formulation is safe. Typically, the product formulation consists of intended ingredient and impurities. I can go into detail. So let's talk about step-by-step approach for risk assessment. The first step, you remember, hazard identification. And you have a new ingredient, new technology. The first step is you need to understand whether this new ingredient or new chemical is dangerous or not. How? You need to assess all toxicological endpoints, systemic and local toxicity. So you need to really understand, is there any adverse event or adverse effect caused by new chemical? How we do that? This is how we do that. The first, before you generate any kind of safety data, we try to understand what are existing safety data so that we can maximize available data without generating data. That is the first step. And then we use a lot of innovative methods. For example, old days, we used to conduct animal testing, old days. These days, there are a lot of scientific criticism about validity of animal testing because animals definitely different from human. And of course, we have a lot of pressure, social pressure for animal welfare. But more importantly, scientifically, we need to really understand toxicological mechanism of a new chemical or new technology, which is more relevant to human. For example, we can use human-derived cell system to really understand hazard profile or potential toxicity of new chemical instead of conducting animal testing. That will provide us more meaningful safety information. Let me give you an example. I just hide the name of chemical. Let's assume chemical A, ingredient A. And then we assess all toxicological endpoints. And then the final conclusion from this step was, yes, there is a potential toxicity of this chemical. This new whitening active ingredient, it causes liver toxicity. It sounds scary. Yes, it's dangerous. That was our conclusion. The second step is the potency. We know this is dangerous. How dangerous it is. So we need to understand the potency and also you remember there's always uncertainty when you extrapolate experimental data to human use scenario. This is how we do. In real world, the chemical, any kind of toxic chemical may cause several different adverse effects. That means not only liver toxicity, but also other toxicity, such as reprotoxicity or liver toxicity or kidney toxicity, you name it. So therefore, you really understand what are real critical adverse events when you conduct risk assessment. How? This is how we do that. The first, you try to understand what kind of toxicity caused by this new chemical. That's the first step. And second one is, you need to really determine potency of identified toxicity based on lowest dose that cause toxicity. I can give you more explanation in next slide. What does it mean by that? This one, critical effect. The definition is very important that when you conduct safety testing, in general, we need to test several different doses. Low, medium, high, for example. And then you need to understand, you need to see some effect. So that's why we use high dose. And then the medium dose, typically we try to see some dose response. In other words, at high dose, you can observe very toxic effect. At medium dose, you hope you can observe some less toxic effect. And then the low dose, hopefully you cannot see, you do not see adverse effect. That's typically we design any kind of safety study. That is a dose response study. And then going back to critical study, we always try to understand what is the lowest dose that see adverse event. This graph is easy to understand for you. Let me give you an example. The y-axis is the severity of adverse effect. And the x-axis is different dose, low to high. And the same chemical cause 3 different toxicities: kidney, liver, and developmental toxicity. See, the same chemical causes 3 different toxicity. However, the lowest dose cause observed toxicity is different. In case of developmental toxicity, that was 200 milligram per kilogram per day. Liver, 100, and kidney is a little over 19 milligram, but below 100. Therefore, in this case, kidney toxicity is critical effect. When we conduct risk assessment, it makes sense because we can focus on kidney toxicity, right? Because if we can cover this kidney toxicity, then automatically cover liver and developmental toxicity. Very straightforward, right? Very easy. And then let's move on to dose response assessment. Okay. There are several aspects when you extrapolate experimental data to human use scenario. The first one is intraspecies. In other words, responsiveness to toxic chemical between you and I may be different. There's always sensitive population. There's always subpopulation who are more vulnerable to the same chemical. So that is intraspecies variation. The second one is animal is different from human. That is interspecies variation. How about the duration? When you conduct safety testing, you cannot conduct 70 years to cover chronic exposure, right? So therefore, let me give you an example that in case of lead, the lifespan is about 2 years. So therefore, when we conduct chronic study using lead, we treat lead for 2 years. Why? Because that 2 years of lead is comparable to 70 years of a human. Going back to uncertainty, think about there's uncertainty because test duration is different. Therefore, we need to consider that aspect as well. And there are many other aspects. For example, if you have a limited safety data, then you want to be more conservative. How about the matrix effect? what if your product formulation contains certain chemical that facilitate skin penetration of your new chemical, then that is not good for consumer safety. So you need to also consider that aspect. There are many uncertainties that you need to really understand. That is part of those response assessment. And this is very nice summary that what I said. Again, when you generate experimental data for hazard identification, you need to understand the potency of hazard, which is dose response assessment. How you need to understand critical effect. And then at the same time, you need to understand the uncertainty to establish safe range. And this is specific example that how we did that. That new chemicals, we found 19 milligram per kilogram per day as a novel, no observed adverse effect level. And then we apply uncertainty factor to establish safe limit. Next one is we need to understand consumer exposure. So we need to understand how consumer will be exposed to new chemical. This is how we do that. So many times we generate consumer habit and practice data. For example, we recruit test subject, and then we gave them test product. And then we asked them use the product as they wish. And then we can measure how much product they use, whether how many times per day, and how much product they are using, and how often they are using. Because when we distribute the product contains the sensor, that sensor collect that information and send us online. Then we can easily analyze those data. And then when we do exposure assessment, the relevant unit is very important. For example, skincare product, you apply the product on your skin. In case of skin allergy, the unit dose we consider is gram per square centimeter. From safety point of view, it doesn't matter how much product in certain skin surface. That is very important concept. How about the air freshener or inhalation type drug, asthma drug, for example? The unit is gram per cubic meter in air concentration. How about the oral injection? It should be gram per body weight kilogram per day. So we always try to use consumer relevant unit. And this is a specific example how we quantitate consumer exposure to new chemical. In this case, we used shower gel. And we know that how much consumer use this product. And then this one is rinse off product. In other words, there are two different types of cosmetic product. One is rib-on, which means you apply on skin and then never rinse off. Just stay there. On the other hand, in case of shampoo or body wash, you apply and then you rinse off with water. So from exposure point of view, we need to focus on how much chemical will be remain on skin after rinsed off. That's what we considered. And then you can use body weight depending on target market, and then we can quantitate consumer exposure. And then this is the final slide of risk assessment, risk characterization. Very simple. Based on experimental data, 19 milligram per kilogram per day, you apply uncertainty factor 0.19, and then you quantitate consumer exposure to this chemical. Final conclusion is consumer exposure is in the safe range. Therefore, we can say this new active can be safely used in shower gel. Everybody follow me, right? It's very simple, very easy to understand. And then, as needed, we also do safety assessment as a formulation, as a mixture of chemical. This is typically we do, for example, skin irritation or eye irritation. How about the package? For example, when you have skincare product package, but what if it has very sharp edge that causes skin cut? Or what if it has very small piece that may have choking hazard? So we need to consider those aspects as well. And then, as needed, we can also put cautionary labeling. For example, avoid contact to eyes, and then if happen, immediately rinse off with plenty of water. So as needed, we can put cautionary statement on the product package. And then we do not stop there, and we keep monitoring any kind of health-related adverse event happen as a result of use of consumer products. If you take a look at cosmetic products, there's always 800 phone number or any contact information so that consumer can contact company or government if they have any kind of health issue. This is very critical information. And in our organization, every month, I get the monthly report, summarize any kind of adverse event and the frequency and severity. We all get those information. And this is a summary of what I said so far. Before I move to next one, do you have any questions before I move? Okay. Very straightforward, right? Very easy to understand. Okay, let me move on to the next topic. That I explain about the risk assessment principle and how we apply those risk assessment principles to evaluate safety of consumer product. But risk assessment principle or risk assessment approach have been continuously refined with the advancement in safety science. In old days, we rely on animal testing. It's like a black box. We just treat animal with very high dose of chemical, and then we kill animal, and we take out organ. And then we weigh weight of organ or histology. Very crude. The problem is that we have to use very high dose that is not really relevant to human exposure condition. Unless you treat animal with very high dose, you never be able to see any toxicity. That's why they use very high dose. The problem is that kind of toxicity study is not really relevant to human. So therefore, there have been a lot of progress in developing innovative methods. That's what I'm going to talk about. This innovative method is highly dependent on mechanistic understanding of toxicity. So if you understand toxicological mechanism of new chemical that cause adverse event, and then you can better be able to assess toxicity, which is more human relevant. And the new approach methodology is kind of generic term that any kind of method can be in vitro or any kind of information allow us to assess toxicity of new technology or chemical by avoiding or reducing the use of animal testing. That is new approach methodologies. And the read across is one of these methodologies. Read across is a very simple concept that we have been using many, many years in industry. You have a new chemical and you don't have safety data at all because it's new to the world. The first step is how we can maximize existing safety data. The first step is, yes, there is a correlation between chemical structure and potential toxicity. It's a well-known fact. Therefore, as a safety expert, I may look for any chemicals which is similar to your new chemical. That is analog. It can be based on chemical structure. For example, any kind of chemical with aldehyde group can be correlated with skin allergy. Any kind of functional group which is chemically reactive, there's a high chance to cause toxicity because in order to cause toxicity, skin allergy, chemical such as heptane can be very small molecular weight, but that become immunogen or become allergen. That small molecular weight chemical must bind endogenous protein. So in other words, chemical should be chemically reactive, active. So therefore, we can get some idea about how we can effectively identify analog. And then, if those analogs based on structural similarity or biological similarity, if they have safety data, then we can use those safety data, reapply to your new chemical. That is read across. Follow me? Concept is very simple. This one is the just general workflow of how we do more systematically. For example, you have a new chemical, and then you need to characterize chemical property of a new chemical. And then it's a hypothesis-driven exercise. You can guess what kind of toxicity may be associated with your new chemical based on characterization of your new chemical. And then you look for analog, and then you use those safety data derived from analog, and then you justify why you can use safety data derived from analog. Why? Whether they share commonality in terms of toxicity. And then you can make a conclusion. And we published several papers working with government regulators. This is more focused on the chemical registration. When you register chemical, you have to submit safety data. And then what we did previously was focus on Korea, Japan, China, try to understand what's the current status of using this read-across approach. Each country has a different stage of applying this read-across approach. Of course, in Europe and North America, it's pretty commonly used, this approach. However, in terms of using this read-across application in terms of regulatory decision making is different. For example, in China, they don't accept this read-across approach. Basically, they are saying if testing is not technically feasible, then as a last resort, you can use read-across application. In other words, they don't accept. That's what they mean. In Japan, they are using this methodology as a screening purpose. In Korea, they are accepting read-across approach for chemical registration. However, very limited, very stringent criteria. And then we work with regulators in Korea and also other stakeholders in academia, professors, and industry, really understand, really discuss what kind of technical challenge associated with read-across approach so that we can use this approach for regulatory decision making. And then several areas we identified. And number one is still we need scientifically strong, robust justification for analog identification. Why analog provide biological similarity so that we can use their safety data. Technical justification for similarity. And then there are a lot of subjective judgment when we do read-across approach. Is there any way that we can standardize this approach so that we can get more consistent outcome? And then another technical challenge is inexperience of stakeholders, in this case, regulators. This is relatively new technology, so that regulators, they feel not very comfortable because they are accountable for their final regulatory decision. They want to really make sure that there's no safety issue at all. And industry is also, they need to learn more. They need to increase the technical confidence. So there are several technical challenges that we discussed. And our goal was, we tried to identify what kind of technical challenges out there, and then what we can do better. What are interim solution, and what is long-term solution, so that we can make a science-based regulatory decision making. That is our ultimate goal. This kind of technical collaboration really instrumental in terms of transparency. Let me move on to risk communication and trace chemicals in consumer products. Now the analytical technology is highly advanced that you can measure any kind of chemical at very, very low level. Let's say ppb level. What does it mean by that? Many cases, many consumers very anxious about safety of consumer product. If news media or newspaper publish articles showing that skincare product contains formaldehyde, which is carcinogen. It's very sensational, emotional. That kind of anxiety we call chemophobia, the fear of chemical. As a government regulator point of view, they're also very concerned about that anxiety, which is chemophobia. At the end, we want to make a science-based safety decision, whether this is really dangerous or safe. What we did was we collaborated with safety communication expert at the ETH in Switzerland. And we tried to understand what can be done to mitigate chemophobia among consumers. What if we provide toxicology information? What can be done? This is what we did. In Korea, we recruit test participants, about 600 people. And then the gender and age and the regional distribution of all participants should be comparable to Korean population. And then we provide very basic information, hypothetically. Here's the shampoo product. It may contain very low level of trace chemical as an impurity, which is technically unavoidable. We provide this kind of information. And then we ask several questions. Let me show you the video, and then we show this video.
In this video, we will talk about chemicals. Nature produces them, and so do humans. Both are chemicals. Most people believe that man-made chemicals are dangerous to human health, while chemicals of natural origin are safe. But that is not correct. The origin of a chemical, whether from humans or from nature, tells us nothing about how dangerous the chemical is. Why is this? Let us take, for example, mushrooms. If natural chemicals are safe, then all mushrooms should be safe. But there are naturally occurring chemicals such as arelanine, found in certain types of mushrooms, that are very toxic, making these mushrooms poisonous to us if we consume them. Another example is ascorbic acid, which is vitamin C, that can be produced by humans in laboratories in the form of tablets and is safe for consumption. Therefore, just because a chemical is natural does not make it automatically safe, whereas a chemical that is man-made is not necessarily dangerous. A central principle in toxicology is that the dose makes the poison. This principle indicates that a chemical is not per se harmful to health. The harmfulness of the chemical depends on the dose that someone is exposed to. Let us take, for example, formic acid. Formic acid, which is found in fruits such as kiwis or is produced in laboratories, can be dangerous to our health if we are exposed to high enough doses of it. However, eating, for example, 2 to 3 kiwis a day is safe, whereas eating more than 200 kiwis a day can be harmful for us. Any chemical can be safely used as long as the dose is within its safe range. So how is a chemical's safe range established? Small amounts to very large amounts of the chemical are tested. The largest amount tested that does not cause any harm is the safe limit. However, this amount is many times greater than what is actually used in a product. An extra wide margin of safety is added for even more protection to establish a chemical's safe range. The amount of the chemicals that are intentionally added in a product or chemical impurities that are unintentionally found in products are always within the safe range. Therefore, being exposed to small amounts of a hazardous chemical in products does not necessarily mean that they are harmful.
Okay, so we showed this video and then we asked the question. So this video contains the three key information. The first one is a basic principle about risk assessment. The second, synthetic versus artificial chemical. In general, consumers think natural ingredient is safe, but that's not true. And then those makes poison. So we provide those three key information.
In this video,
This is the test result. We ask consumer acceptance of trace chemical in consumer product, whether they feel more comfortable after watching video. So we compare before and after watching video. And then if one step further, we check purchase intention. In other words, are they willing to buy consumer product containing trace level of chemical or impurities? As you can see, the acceptance increased, willingness to purchase also increased. So therefore, this kind of informational video may be useful to further mitigate chemophobia among consumers. This is my real final slide. So as you can notice that I intentionally repeat several key concepts over and over to make sure that you internalize key concepts. When we develop a new technology, let's say whitening active chemical used in skincare product, the first, we use risk assessment principle to evaluate or to characterize risk of your new technology. And then, once we successfully launch your new chemical or new technology used in skincare product, for example, we have a post-market surveillance system to make sure that our risk assessment was correctly conducted. As needed, we can go back and we can reformulate as needed. And this risk assessment has been continuously refined with advancement of safety science that allow us to conduct more comprehensive safety assessment. And finally, risk communication is critical. Collaborative effort of all stakeholders in risk communication is very critical. It's key to ensuring safety of consumer products sold in the market. Thank you for your attention.
Okay, yeah, thank you for the insightful talk. Yeah, Dr. Soga. Yeah. Now I would like to invite the audiences and participants for the question and answers, and also for those who are in Zoom, they can ask question or discuss by using the chat features that is provided in the Zoom. Yeah, so I will invite the audience to open up the question and answer session. Yeah, please do. Yes, please. We can pass the microphone. Yeah, maybe you can state your name as well.
Check. Okay. Hello, sir. Kwon. My name is Dandy Cristanto. I'm from Pharmacy 2019. First of all, I want to thank you for the presentation. It was really insightful for me. I want to ask. So you mentioned before that each component in the ingredients should be assessed by its safety, safe range, right? And then, but what if, for example, you want to make a shampoo with a new chemical? For example, like surfactant A, primary surfactant A. So this surfactant is actually really really critical for the quality of the shampoo. So for example, the optimum percentage of the ideal percentage of the surfactant, for example, 10%, but your new chemical at that 10% is not meet the safety range of the criteria, right? So what would you do? I mean, would you like find another ingredients, or lower the concentration, or changing into alternate ingredients with lower quality, or what would you do? Thank you.
Okay, so your question is let's say hypothetical, there's a new surfactant, and then there's a conflict between efficacy level. In order to have a good efficacy, let's name 10% is a minimum requirement. On the other hand, based on safety assessment, 5% is safe limit. What should we do? The first is that depending on what kind of safety concern, that's the first one I would do. This is my subjective answer. The first, that 5% of safe limit is based on what? I would be very cautious about carcinogen. If it causes cancer, out. It's not worth it. Number two, reproductive and developmental toxicity. If your new chemical causes birth defect, out. It's too dangerous. However, if you are concerned about local toxicity, skin irritation, or eye irritation, then I will generate additional safety data. For example, at the ingredient level, if you test 5% of that surfactant only versus how about the matrix effect in the formulation? For example, there's a paper published showing that non-ionic surfactant may mitigate potential irritation profile of anionic surfactant. I would play around with different combination of different type of surfactant in the context of formulation. As long as your safety concern is local toxicity, skin irritation, eye irritation. Typically, surfactant is usually the point of contact irritant. It does not cause cancer. It does not cause reproductive toxicity. In general, it causes irritation or corrosion. Therefore, there is a high chance that you can refine safety data using different combination of different ingredient. Final suggestion is I would do in vitro study using different surfactant type, anionic, non-ionic, cationic. Usually we don't use cationic. Cationic is very toxic, used as anti-active ingredient. I would say non-ionic and anionic surfactant, different concentration, generate data, and then we can develop model. We can predict different combination of non or anionic surfactant irritation profile. And then once I get some idea about what combination provide acceptable irritation profile by using market product as a benchmark. Because shampoo, there's always irritation concern. It's impossible to have shampoo does not cause irritation. It's impossible. You always have risk-benefit argument. Therefore, you further refine safety data, irritation data, and then risk management approach. For example, you can use safety cautionary statement to reinforce that close your eye or avoid eye contact. So I would use those two approaches.
Okay, hopefully it answers your questions, right? Yeah, so before I will pass the next question, so for those of you who wants to participate in filling the cost evaluation survey, right? You can do so by scanning the screen, yeah, or you can type in the page, right, and fill in the evaluation score, yeah, for the rest of you. All right, and I would like to open up another questions. I see you are about to raise your hands. Yes, please. Yeah, introduce yourself and you can proceed ahead with the questions.
So my name is Joshua from Biotechnology 2019. So lately there is an issue about ethylene glycol in the cough syrup. Is there any justification for the usage of ethylene glycol in the common cold medication? Thank you.
So your question is ethylene glycol. And that is a pretty toxic chemical that we should not use as an intended ingredient. So what was your question?
So lately there is a case about the usage of ethylene glycol in the cough syrup in Indonesia. So there is the damage is already being done to the mostly child below 5. So it caused the chronic kidney disease, and maybe I want to know is there any justification if maybe ethylene glycol is being used in the manufacturing of the cough syrup or common cold medication, something like that?
I would not recommend to use ethylene glycol. It's a well-known kidney toxicant.
Okay, thank you for your answer.
Okay, so I can see several students are about to raise their hands. Yes, please. Next question, please. Can you raise your hand so that I can see it? Yes, please. Yeah. Okay, introduce yourself and state your questions.
Hello everyone, my name is Ilen Salmasyara. I'm from Bioentrepreneurship Cohort 2018. So actually I'm a little bit confused that what are the importance for the dose response assessments to build a relationship in the toxicology itself. So yeah, that's all my question. Thank you.
I don't understand exactly. Can you be more specific?
Okay, so I'm still a little bit confused about what are the relationship between the response assessment itself with the toxicology. Thank you very much.
So you want to better understand the dose response assessment?
Yeah, the relationship between the dose response assessment itself with the toxicology. Yeah,
Okay.
so that's all my question. Thank you.
Yeah, let me try because let's say the dose response is when you have a new chemical and then your new chemical cause liver toxicity, for example. And then you need to understand the potency of liver toxicity because your goal is you want to establish the safe range because those make poison. When you generate safety data, you want to test your new chemical at different dose. High, medium, low, for example. And then you want to see some liver toxicity at high dose. And then at the medium dose, the severity of liver toxicity may reduce. So that means dose response. And then hopefully at low dose, you do not see any liver toxicity. That is no observed adverse effect level for liver toxicity. So this we called dose response assessment. Did I answer your question? You can come back to me. You can send me email anytime. Then I'm happy to give you more explanation. So everybody feel free to send me email. You have my email address. That I also provide coaching and mentoring for young people like you. So any kind of technical question or career discussion, you can reach out to me.
Yeah, thank you. So I would like to request from this side, probably. Yes, please.
Okay, thank you, sir, for the presentation. I am Aurelie Rose from Pharmacy 2020. So I want to ask, basically, you mentioned like there are some uncertainties, right, when we are like doing the risk assessment, like determining concentration at which the product or chemical gives the toxic effect. So how can we be sure, like, when the evaluation is certain? Like, how do we deal with the uncertainties? Like, yeah. Thank you.
So your question is about uncertainty, how we deal with uncertainty when we establish the safe range.
Yes.
Okay. In my talk, I provide several aspects we need to consider about uncertainty. For example, inter-individual variation or intra-individual variation. For example, you and I, we are exposed to the same chemical. However, toxicokinetics and toxicodynamics may be different, right? Because your body, how your body deal with new chemical, once new chemical get into your body, may be different from mine. So therefore, when you generate pharmacokinetic data and the pharmacodynamics data, pharmacodynamics means what new chemical does to your body once new chemical get into the body. In other words, what kind of toxicity caused by new chemical. And you and I may respond differently. So therefore, we can generate toxicokinetics and toxicodynamic data. Those data can cover inter-individual and intra-individual variation. How about the duration? Your experimental condition is a very short-term testing. However, skincare product, for example, consumer use for 70 years, for example, for the rest of your life. And then how you can make sure that those uncertainty, because your experimental data derived from very short-term study, very short-term. Depending on toxicological endpoint, for example, skin irritation. Skin irritation is point of contact irritant. That means duration doesn't matter. If you are exposed to very strong skin irritant on your skin, you have a problem. It doesn't matter you are exposed to once or continuously for the rest of your life. Local toxicity such as skin irritation or eye irritation, duration doesn't matter. How about the product matrix, your product formulation? What if your product formulation contains certain solvent that facilitate skin penetration of your new chemical? That may increase potential skin allergy safety concern because small molecular weight chemical called heptane have to go through the stratum corneum of your skin to become allergen. So therefore, depending on the product matrix, what kind of ingredient present in your product formulation, what kind of uncertainty associated with product matrix, you can understand. So to answer your question, by understanding toxicological mechanism, you can reduce uncertainty. And nonetheless, you can also apply additional uncertainty factor to be more conservative and to be more safe. So
Thank you.
that is current practice.
Thank you, sir.
Okay, I think there are already our next questions.
Okay, hello. Thank you for the informative talk. My name is Patricia from Pharmacy 19, and I would like to ask regarding the current testing alternatives for animal testing. So currently, is there a way for a systemic toxicity testing using in vitro or other animal models? Because like especially in obtaining toxicodynamics and toxicokinetics data, is it feasible to not use animal testing in the future? Thank you.
So your question is there any way that we can completely avoid animal testing? That is our ultimate goal. And in my talk, I provide new approach methodology. That is exactly what we are currently working on. There are several ways. Computational toxicology, in other words, based on chemical structure, you characterize chemical, and then we call chemical profiling. If you understand chemical structure, and then you can predict potential toxicity. And then how about the toxicogenomics? If you understand gene expression as a result of, for example, you treat human-derived cells with new chemical, and then you understand gene expression profile of that cell as a result of exposure to chemical. And then you can correlate those gene expression trend with potential toxicity, because we already have benchmark chemical we know certain chemicals are known to be carcinogen or known to be liver toxicant. So therefore, we can compare gene expression profile. Read across. You have a target chemical that you can identify analog that may have similar toxicity profile based on biological activity and structural activity. Or another high dose, high toxicity, medium toxicity, low dose, no toxicity. The low dose we call point of departure. In this case, no observed adverse effect level. That is applied dose, external dose, because we don't know how much your chemical get into the liver tissue, right? Therefore, that is external dose. The scientific challenge is when you do in vitro test, that is internal dose, because you culture cells, you treat chemical by mixing with culture medium with your chemical. That is internal dose. So how you extrapolate that internal dose, let's say you conduct high throughput testing using in vitro testing, and then you identified, okay, you treat 10 milligram, 10 micromole, 100 micromole, let's say 10 micromole is novel based on in vitro test. What does it mean? How we can extrapolate to external dose? Because that's very important concept, because when we conduct exposure assessment, consumer, they are exposed to chemical when they use consumer product, that is external dose, applied dose. So therefore, that is very important, how to we extrapolate internal dose to external dose. So that is pharmacokinetic and pharmacodynamic study. That is critical. So as of today, for systemic toxicity, we still cannot completely replace animal testing. However, that does not mean animal testing is better to predict human toxicology. No. Many times, animal testing data does not predict human toxicity 100%. There are literature published that they are saying less than 50% is predictable based on animal testing. So therefore, to go back to your question, it's very critical question that how to avoid, how to reduce use of animal testing without compromising science. That is you and I need to do. That's my answer.
Thank you.
Okay, I think we only have one slot for one questions left. Right, so I think we can invite the last questions.
Hello, sir. Thank you for your presentation. So I am Tiara Angelica from Pharmacy Core 2019. So from what you explained earlier, that the safety assessment consists of many aspects, such as the chemical characterization and then dose dependent assessment, exposure assessment, and finally the risk assessment. And from your explanation, I believe as well that the assessment principle are pretty similar to the project conducted here in ITL, such as what my friends already done, such as the toxicity testing, clinical trial, and etc. Where usually several months are needed to complete the project. Meanwhile, for developing the product that you explained before, that the chemical in its product must have the safety data as well, meaning that the product must undergo all the safety assessment. So here, actually, I'm a bit curious that how usually the real pharmaceutical companies such as the P&G manage the workload and time for developing each product to finally manufacture the product. Thank you, sir. Do you understand what?
Can you repeat the last part?
Oh, so I'm curious how usually the pharmaceutical companies, such as the P&G, to manage the workload and time in assess the safety data to finally can manufacture the product. Thank you, sir.
So how P&G
Manage the workload and time for doing the safety assessment for each product until it finally can be manufactured.
Okay, how P&G can make a final decision?
Yes, and what usually how long is the duration needed for
How long?
developing each product?
Sorry,
Oh, sorry, sir.
so your question is
How the PNG manage the workload and time in doing the safety assessment?
okay, how much effort
Yes.
P&G put into safety assessment?
Yes.
In terms of time and
workload.
Workload?
Yes.
Okay.
Thank you, sir.
I don't have answer your question. It depends because depending on the product type, there's a product development cycle. It can be 5 years or 10 years because we have a certain technology platform that very important, but it can be reapplied to many different product types. There's more focus on basic understanding of science. It's a long term. And then on the other hand, the other product project can be very short term. It can be 1 year. Or many times we can change very minor modification of the product. For example, shampoo, if we just change the perfume, it can be quick. So it depending on what kind of product, what kind of technology, we customize how much effort we need. So it can be very short, it can be very long term. And then the test method development can be very long because you develop new test method and then you need to get endorsement from scientific community through publication and also by regulator. For example, OECD validate test method. When we submit, we need to chemical registration. We need to generate safety data for new chemical. We have to use certain test method. We cannot just use our internal test method because other people don't trust. So therefore, there's a OECD externally validated test method to be consistent globally. If we want to develop that kind of test method, it may take 10 years or 20 years.
Oh, so it's similar to the drugs?
Hmm?
So it's similar to the drugs
Yes.
to
Yeah.
produce the medicine?
Yeah, it depends. Even the pharmaceutical drug, it can be very quick to screening stage. When you screen candidate technology, it can be very quick. However, if you want to register, you have to conduct a lot of safety testing and phase 1 to 3, and even post-market surveillance. If you include everything, it may take many, many years. So depending on product type and what kind of project, it varies. It can be short and long.
Okay, thank you so much, sir.
Okay, so I believe the time is up, right? So I would like to remind participants, both here and the Zoom, to fill in the session talk evaluation presented by the QR code in the screen. We appreciate your time to do so. And after this, we will have a photo session and certificate of appreciation. So please do not leave the room yet. Okay, so we would like to thank you for our honorable guests. Please give a round of applause. To Dr. Seok Soga Kwon. So here is our humble certificate of appreciation. So we can take a picture.